Okay, then another story. There are about 15 stem cells in each crypt, nicely confined. We still have 15, and we wanted to study them independently. This was done by two PhD students in the lab that have both graduated who were allowance. They made this construct here. So the previous Curie reporter turned the stem cells blue, but if we would mark 3 stem cells, they would all 3 be blue, and it would be difficult for us a few days later to see who was who. So they actually constructed a reporter that would let a stem cell randomly pick, and I won't go into the details, any of 4 colors. And when you make it homozygous, any of 10 false colors, and you draw the checkerboard. So when a mouse has this construct in the genome, and we activate Cree, every cell picks its own color, and this can be then green, yellow, red, or blue. It essentially rearranges these pieces of DNA. And the outcome that we hope to see was the following. So we mark all of the stem cells in different colors, and then we just sit and we wait, and then we should see over the next few days that the crypt gets filled up with cells of different colors. And what we noticed is that slowly one of the stem cells was winning. And after about 2 months or so, the crypt had entirely a single color. And I won't go into the details of that. And this is what we hope to see, that eventually, so this is a live mouse, we would actually see this sort of image. And that actually looks maybe a little exaggerated, but these are the images from the mice that we get. So this is again live tissue. Yeah. So this is maybe a bit different. See, this is one crypt. There are actually several crypts that surround one village. And we had predicted that the bands that come out of the crypts would move in parallel bands to the tips of the villi. That's exactly what you see. This is one large village here with a number of crypts. So here the red stem cell has one, there's a blue stem cell here, red, and in between it here there's a yellow and a green one. And they nicely produce these bands of cells. And for the rest of the lifetime of this mouse, it will look like this on its inside. It doesn't know it until you look at this one. Okay, so now in the last bit of the talk, we'll actually use this construct for we hope to publish this paper in the next few months, to address whether there are stem cells in cancers. And I should explain a little bit here. My lab has worked for a long time on colon cancer. And colon cancer is a good model to study cancer because typically it starts with the loss of a single gene, APC. The APC gene is almost in every case of sporadic or hereditary colon cancer. This is how it starts. When you lose the APC gene, both alleles, the cell will become an adenoma. And then later on it accumulates more mutations, and in that adenoma the cell becomes more and more malignant, they become invasive and metastasize, and then you have a full-blown adenocarcinoma. But it starts with the loss of APC. And we can model this in the mice because we have a mouse where there are 2 LOX-P sites in the APC gene, and with our Cree enzyme we can at will, we can inject tamoxifen, delete the APC gene and in a single cell, and then turn that single cell into the starting point of a cancer. Hope you can follow this. So a paper we published a few years ago that I won't show here, but if we delete the APC gene in any of these cells, so these very rapidly proliferating cells or the differentiated cells, nothing happens. But if we delete it in the stem cells, we make an adenoma. So this actually showed for the first time that cancers start in stem cells. Might sound very obvious, but that had never been formally described. And to summarize this again in a little movie, so we have a mouse, now it's floxed APC, so we inject tamoxifen, low dose, so in some of the stem cells we hit this APC gene, that stem cell now is a malignant stem cell, and then we can just look over time what happens, and hope this plays, yeah. So we see that this malignant stem cell will kick out the other stem cells that are there very rapidly, and in the meantime it makes daughter cells that you can see here in brown. They look pretty normal, like the ones on the right, but then when they try to differentiate out, they cannot do it, and they continue to proliferate because they don't have this APC gene that they need to differentiate. And over time, it takes about 30 to 40 days, then the mice get very sick. They make these adenomas, these polyps as they're called, that are then in human life, when you have these and you remove them, you're fine, but when you leave them for 10, 20, 30 years in your colon, additional mutations will occur in single cells and they will become adenocarcinomas. Now, we did this and we noticed that in these adenomas, there are LG5 stem cells, so it looks like the cancers have their own stem cells. And again, they are flanked by dependent cells, much like what we normally saw in the normal crypts. And this is how we show this. So these are two normal crypts, and the sizes here, magnifications here and here are the same. So the stem cells are in green and the dependent cells are in purple. So this is what you would normally see in an adenoma of, say, a month old. Although it looks very chaotic, but if you look carefully, you can see the same architecture there. There's a green cell, a purple cell, green, purple, green, purple. So the adenoma is like a caricature of a normal crypt. And there's been a lot of speculation over the last decade that in tumors, in cancers, 99% of the cells are not so bad, but it's the stem cells in the cancers, cancer stem cells, that are the ones that if you treat the patient and you shrink the tumor to almost nothing, they survive and they will take care that the tumor grows back. And as people have believed for a long time, that's the reason when you cannot radically do radical surgery and remove the entire tumor, but when you irradiate, you give chemotherapy, that you can get great initial results. Tumor is invisible, but as you all know, when you treat with chemotherapy or radiation, you very rarely cure the patient. It will always rebound the tumor. And it was believed for a long time that there are stem cells, rare cells that sit in the tumor that are responsible for this response. So here we think we actually, in the mouse, we have them, so they're green, and they're still apparently dependent on the purple dependent cells here. And what we wanted to do in an experiment now is, I hope that this works again, so cross our APC Flox mouse with the multicolor reporter. This gets a little complicated. People come in through all doors, I know. So delete APC, but at the same time, in the same cell, we give the cell a color, a random color, and then if a tumor starts growing from that stem cell, that tumor will have its own color. Right, let's see, you see it happening here, so the stem cell lost APC, so now a tumor stem cell, but it's also red. And if the tumor grows out, it should be red. Now that was the idea, and indeed this works. This also works. So this is actually, there are here 5 tumors in a row in these mice. So the muscle layer is here. These are the villi that are deleted. But this is one tumor starting from a single stem cell that picked the color red. And here's another tumor from a single stem cell that picked the color yellow. Now, the red tumor cells, the reporter that originally had 4 colors still has 2 colors left. And now we thought, well, if we now let these tumors grow, and now we again induce the creativity, we can possibly switch, and you can see it hopefully here. So this is the tumor with LD5 cells. In one of those LD5 cells, we can flip the red color to blue. And this can only happen in the stem cells in that tumor. So then we have a blue cell in a red tumor, and now we can just follow what this particular cell, how it behaves inside that red tumor. It is part of the tumor with systems. So I hope you can follow this. And this actually shows over time, so it's very chaotic. The images are not as nice as if you get a normal crypt. But indeed, so we induce tumors in stem cells, and the stem cells and the tumors have their own colors, as you can see here. So this is a red tumor. We wait 24 days, so the tumors are fairly large. Then we induce the flip of the colors in the stem cells in the tumor. And then we just follow over time what happens. So 2 days after we do that flipping, the retracing, we get 2 or 3 cells here. By day 6, there's already more offspring. By day 9, you can see there's a large, particularly here, there's already a large part of the tumor now derives from that cancer stem cell. And if you look here on the left, we look at the number of cells per clone, so it starts with 1 when we induce a single stem cell in a tumor, but then very rapidly we go up to 60 to 80 cells that are produced by the single stem cell. And then when you look at the number of stem cells in one of those clones, you also go from 1 to quite rapidly to about 20 or so, 10 to 20 of these stem cells. So this, probably complicated to follow, but what this shows is that, first of all, colon cancer starts in a single stem cell by the loss of APC. When the tumor grows, it has its own stem cells that are marked green by LD5, and those stem cells are really the fuel of the growth of the tumor. So a lot of the other parts of the tumor do proliferate, but many cells are lost, but it's the green stem cells in that tumor that take care that these tumors are not in homeostasis, but they continue to grow, and eventually will kill the mouse. And with that, I'd like to end by naming a few of the people that were involved in all of this. Nick Barker Here is the postdoc that found this Altair 5 marker. That's really a fantastic tool. And I should mention in particular Laurent and Hugo, who made this multicolor mouse, the confetti mouse as it's called, and Toshisato, who is the Japanese guy who did all this, who set up all these beautiful filter systems. And with that, I'd like to thank you very much for your attention, and I'm not really sure what we're going to do now.